Pain is the commonest reason that a patient presents to their primary care physician, and yet we have very poor alternatives to try and treat them. The commonest analgesics are either non-steroidal anti-inflammatory drugs or opiates. And in the case of the opiates, we have an enormous crisis with opiate overdose, diversion, and abuse. There is therefore an urgent need for the development of novel analgesics that are as potent as opiates but without their abuse liability. And this is something that we are devoting considerable effort to. And the question is how to do that. The major problem is to identify a target that is completely distinct from the mu opiate receptor, which is the receptor, the GPCR, that opiates act on. And the approach we have taken to do that is to use human genetics as our starting point to try and identify completely novel targets or pathways that are involved in the pain process and that provide an opportunity for the development of completely new classes of analgesics. This is what we've done. We'll tell you something about how the human genetics have identified a novel target, and then the challenge is how we can use mouse genetics to identify the site and mechanism that the pathway is involved in the generation of pain. And having done that, we can then begin to test in the mouse the application of new tool compounds that act on that target and see that as being the starting point for the development of completely new analgesics.
This study is based on results that we previously published in late 2006, looking at the regulation of GCH1 after nerve injury. We found that there was a massive upregulation of GCH1, which we saw at the protein and the mRNA level. And it turned out that the product of GCH1, which is BH4, was also very heavily upregulated in the injured neurons. The reason that BH4 is important is it's the essential cofactor for the production of dopamine, serotonin, norepinephrine, epinephrine, and all nitric oxide through the NOS enzymes. In addition to proving that this dysregulation actually caused neuropathic pain, we also found a human haplotype that interfered with the regulation of GCH1 after nerve injury.
First, we used a reporter line to characterize the different cell types that engage the BH4 production pathway after peripheral nerve injury. And we found that both DRG neurons and macrophages, which infiltrate the damaged nerve, produced increased levels of BH4. This discovery that macrophages produce BH4 suggested an unexpected role of this pathway in inflammatory processes. Next, we used gain and loss of function strategies to validate the role of BH4 in pain sensitivity. We used tissue-specific inducible transgenic lines that can either overexpress or are incapable of producing BH4 only in sensory neurons. BH4 overexpression led to an increase in pain sensitivity, whereas its absence did not affect normal responses to noxious stimuli. Genetic disruption of BH4 production only in sensory neurons, however, did reduce and reverse existing neuropathic pain symptoms after nerve injury. To test if a systemic pharmacological inhibition of the BH4 production pathway could reduce neuropathic pain symptoms, we targeted the enzyme Sepiapterin Reductase, SPR, the last enzyme in the BH4 de novo synthesis pathway. This enzyme is a much more druggable target than the other members of the pathway, because it has a distinct and accessible pocket site. Using structure-based design, we developed a potent SPR inhibitor to better fit in this active pocket, and we confirmed this by analyzing a co-crystal of our tool compound in the active pocket of the SPR enzyme. Next, we tested its inhibitory activity using both cell-free and cell-based assays. This new compound is a useful tool to test the physiological and pathological role of BH4. Systemic administration of this SPR inhibitor significantly reduced BH4 levels in DRG and sciatic nerves after peripheral nerve injury, and it's also significantly reduced pain symptoms by acting through neurons and macrophages. This treatment also reduced pain in inflammatory pain models, showing that the BH4 pathway contributes to diverse painful conditions. A key factor when developing potential new analgesic treatments is the ability to reduce pain symptoms without causing adverse effects. Repeated administrations of our compound did not cause any observable side effects. This is because when SPR is blocked, or even absent, cells can still produce low levels of BH4 through the salvage pathway, therefore preventing side effects caused by global BH4 deficiencies. This salvage pathway involves the production of a metabolite called sepiapterin, which is only produced when the enzyme is blocked. We found that sepiapterin accumulation represents a reliable biomarker for SPR inhibition, both in vitro and in vivo. Such a biomarker could therefore represent an extremely helpful tool to titrate the enzyme inhibition to reduce BH4 levels and pain symptoms without causing any side effects.